Hi, my name is Reagan Bushok, and today I'll be presenting the isolation and characterization of the lytic phage toreador for my final presentation in ABE 226. Just a quick introduction about myself. I'm a sophomore in biological engineering with a concentration in cellular and biomolecular engineering with a minor in biotechnology as well, and I'm an undergraduate researcher in the Brubacher lab in BME. The objectives of this semester's research was to contribute new data to the HHMIC phages database by finding novel bacteriophage in the environment and completing the different protocol of direct isolation, purification, all the way to DNA extraction to characterize the phage and be sent in for sequencing over winter break. Right now, studying phage is a very up-and-coming area of research. They are the natural predators of bacteria, but they can be programmed to kill specific bacteria strains, can help us better understand antibiotic resistance, the lytic versus lysogenic phases, and successful phage therapy has already been studied. So it's a very exciting new area of research that we get to contribute by characterizing our phage. So here's a flow diagram of the 6 main protocols that we followed this semester. To start with isolation, we collected an environmental sample and went through isolation and plaque assays to isolate phage from the environment. Looking at my soil sample collection, I collected this sample at the Bruder University Horticulture Garden directly underneath and around the roots of the Toreador coxcomb flower, circled in red. Some of the weather conditions are listed right there, and the soil conditions were dark and slightly damp, maybe due to previous precipitation. Then following the direct isolation protocol, our goal was to extract phage from the environment and complete the protocol to directly isolate it. That plate to the left is the very first isolation of our phage toreador, and the plate to the right is the same plate, except the plaque A is circled where I picked a plaque. And the results from this was that the phage were successfully isolated, and 2 plaques were picked to inoculate host bacteria and proceed to purification. We know we were successful because small clear circular plaques can be seen with cloudy edges around those plates, and those show that phage are present. There are some agar chunks, but those plaques are very clear. Our second step is purification, which we want to purify the phage so that there's only one morphology that we're studying before going on to amplification, so that we can characterize it successfully. The image to the left is after picking that plaque A in the previous slide and doing a sterile dilution of that plaque, and this plate on the left was flooded to collect lysate A. Then the image in the middle is we picked another plaque B and then diluted it for this plate, and the plate was flooded to collect lysate B, and both plates have a lot of clear plaques present, which is a good sign. And then we did a sterile dilution of both those lysates as well for a second sterile dilution to check the morphology and the titer, and it was chosen that lysate B was going to move forward with amplification, and the titer of lysate B was calculated to be 5.1 times 10 to the 7th. And we can confirm that there's only one morphology after comparing plaques from both lysate A and B. The plaques are similar in diameter, size, and visual characteristics. They're very clear circles with cloudy edges, and the titer was not high enough though, so lysate B is going to be used in amplification, which is the third step. So the third step then, after we've purified one morphology of phage, we need to make a bunch of copies of that phage DNA by making webbed plates, which is going to be a plate that is absolutely full of plaques and that has a high enough titer that we can use for DNA extraction, microscopy, etc. Here are the calculations for the titer, the plaques needed for a web plate, and the volume needed for a web plate. And our goal was to create a lysate with a minimum titer of 5 times 10 to the 9th PFE, which is the number of plaques per microliters, and enough volume to successfully use in DNA extraction. This is done by making additional plates with a very high density of plaques, which is that webbed plate I talked about, and flooding those plates to create new lysates until the titer and volume are high enough. However, this step was very difficult for us because we had to do 5 rounds. Our phage was very stubborn with amplification, and we're honestly not 100% sure why. So to start with the left was the lysate B that we originally used. That plate is the first round of amplification. We collected high titer lysate 1 and diluted it to find the titer, and the titer was 5.1 times 10 to the 7th, which wasn't high enough. So we did a second round. The second titer was 1.17 times 10 to the 8th, which was still not high enough. So we did, here are 3 more rounds. You get that third titer on the right, and then we finally found high titer lysate 4, had a titer of 2.3 times 10 to the 9th, which was actually used for DNA extraction, and we did a 5th amplification in our 5th high titer lysate titer actually went down. So that 5th one was used for archiving, and we used the 4th for DNA extraction. We tried a lot of different things for this to try to overcome it, but ultimately we ran out of time, and our phage was being very stubborn, so that is the highest titer we could get, even though it's less than what we needed. Finally, step 4, electromicroscopy, was used to image our phage and figure out what family it was a part of. Here are actual live images of our phage. You can see the small circular round caps with heads and the long flexible tails in both images, and this shows that we have a phage of the Siphoviridae family, which is really exciting. Finally, with DNA extraction, we essentially extract the phage DNA that we can use for characterization. So we went through this protocol, and the spectrophotometer image on the left was used to quantify phage DNA through light absorbance, and our concentration read 231.05 nanograms of nucleic acid per microliter, which means that's definitely a high enough concentration for characterization, which is the next step. So looking at future work and applications, our immediate next steps is that we're going to be characterizing small sections of the phage's genome. We can make genetic fingerprints by restriction endonuclease enzymes. We'll be doing that next week, actually. And we can compare to other phage via gel electrophoresis. Next semester, the archived phage will be sent to C phages for a full genomic characterization, and then that genome will be placed into the database and be used in our bioinformatics course. And looking at then future work and applications, if we were to keep working on this project, we could study bacterial resistance and how phage can overcome antibiotic resistance and phage therapy. You can program phage to attack specific bacterial strains and investigate the potential to target mammalian cells. You can study the lysogenic and lytic cycles to better understand phage infection. You could use genomic editing tools such as CRISPR to alter phage, and you could also study the protein makeup of phage. There's a lot of different avenues that you could continue work with after this semester. Definitely were a lot of problems to troubleshoot and overcome. The main 3 were organization issues. We had 3 lysates at one point instead of 2. We're really confused as to why that was an issue. So, we solved this by sitting down with Lauren and the peer leaders to correctly reorganize everything. We had a contamination issue a few times, and that was solved by reanalyzing why that was occurring and paying special attention to make sure aseptic technique was used correctly. And, we had a lot of problems with amplification. We had 5 rounds and still not a high enough titer. And we tried solving this by doing a bunch of different things, different plaque size calculations, volumes. I went into lab outside of class time to let the flooded plate sit longer, and we consulted with Lauren. So we really tried every way we could to solve this issue. We weren't successful in doing that final titer, but it was a great opportunity to problem solve. We also had some critical decisions that we needed to make this semester. First of all, we had to choose which plaque and lysate to continue with amplification, because there was lysate A and B. During purification, actually, my serial dilutions for lysate A seemed to be backwards, and they didn't make sense. So after consulting with my lab partner, we decided that his serial dilutions made more sense, and we continued with lysate B onto our 5 rounds of amplification. And we also had to decide when to stop amplification, because we could have kept trying to do that to get to that final high titer that we needed. But we tried a bunch of different things, and consulting with Lauren was determined finally that we're out of lab time, and our 4th high titer lysate was high enough for DNA extraction. Definitely with any research project, improvements can be made. If I could go back, I would definitely stay more organized and complete my lab notebook on the same day, pay more attention to aseptic environments to avoid contamination, check my calculations more thoroughly, and also have more faith in my skills and learning ability, because it was my first time in a research lab setting like this, and I was definitely nervous. How is our phage toider unique? What's of the family Siphoviridae? It was found in a very unique location under a very unique flower, so maybe there's some relationship with that if studied further. It was very quickly isolated, but was very stubborn with amplification, so that makes toider unique in itself. And finally, my advice for next year's phage hunters would be to stay organized and complete your lab notebook each week, get comfortable and pay special attention to aseptic technique. Know your protocol very well each week beforehand, because you need to be prepared for each lab to know what you're doing and be successful. Definitely communicate thoroughly with your lab partners so you're both on the same page. Definitely get to know your peer leaders, because they can help you, guide you in the process and give you great tips and advice. And get involved and excited with this class. This is a very exciting class and very exciting research, so be excited about it. Definitely want to say a quick thank you to Purdue University ABE Department, Whistler Center for Electromicroscopy, the C-Phages program, and the Howard Hughes Medical Institute, all those involved in this ABE 226 class, and my lab partner, Michael Hardy. Thank you.